Number 1. Pithovirus sibericum. The zombie virus revived after 30,000 years. In a quiet French laboratory, a small culture dish holds something that should not be alive. Inside that dish, an amoeba drifts through a thin film of nutrient liquid. Around it, particles begin to move, attach, and replicate. Those particles are not modern organisms. They are a virus that last infected a host, while mammoths still walked across northern Eurasia. The virus is called Pithovirus sibericum, and its story begins not in the ocean itself, but in the frozen ground that feeds it. Siberian permafrost works as a kind of biological freezer. It holds soil, plant material, animal remains, and microbes in suspended chemistry. When researchers led by Jean-Michel Claverie and Chantal Abergel of Aix-Marseille University drilled into that frozen archive, they were not hunting for monsters. They were sampling deep layers of Ice Age sediment to see what could still be cultured. What came back surprised them. The team placed thawed sediment with cultures of Acanthamoeba, a common soil and water amoeba used as a kind of bait host. Within days, the amoebae began to die in a pattern that suggested viral infection. Under the microscope, the cause appeared as an enormous oval particle, roughly 1.5 micrometers long. That size matters. Most viruses are so small they require electron microscopy. Pithovirus is large enough to sit at the edge of what light microscopes can resolve. Its genome turned out to be a double-stranded DNA molecule of roughly 600 to 610 kilobases, encoding around 467 predicted proteins. It belongs to a broader group informally called giant viruses, formally placed among the nucleocytoplasmic large DNA viruses. Its shape is described as amphora-like, with an apical structure at one end that appears to guide genome delivery. The term zombie virus is a media label, not a scientific category. Yet the implication is real enough. Knowledge of this virus had been lost in the simplest way possible. It was buried, frozen, absent from the modern ecological record, and unknown to current biology until a sample thawed in a dish. The mainstream theory holds that ancient viruses can remain infective when preserved in stable, cold, oxygen-poor conditions. A more archaeological reading treats permafrost itself as a biological time capsule, where vanished ecosystems leave traces no fossil record can match. A fringe view goes further, suggesting that as Arctic systems destabilize, unknown ancient viruses could emerge into a modern biosphere that has never met them. Warming, mining, drilling, and Arctic infrastructure all disturb layers that have been sealed for millennia. The lab work itself was careful. Replication attempts used amoeba hosts, not human cells, and were conducted as controlled revivals rather than reckless human pathogen experiments. The takeaway is simple. Cold does not erase biological information. It can preserve it with astonishing fidelity. And if a 30,000-year-old virus can wake up in a French petri dish, the next question is obvious. What else is waiting under the ice, and what happens when the ground that holds it begins to drain into the sea? Number 2. Pandoravirus salinus, the giant marine virus with genes biology cannot explain. Follow that question south into the cold coastal waters of central Chile. Off the shore near the city of Tuncan, marine sediment piles up in slow gray layers. To the eye, it is mud. To a virologist, it is a library. In 2013, a research team that included Jean-Michel Claverie, Chantal Abergel, and Nadège Philippe published the discovery of one of the first members of a new viral lineage. They named it Pandoravirus salinus. The reference to the Greek myth of Pandora was deliberate. The container, once opened, revealed biological unknowns that did not match anything in existing genetic catalogs. The host range, so far, is narrow. Pandoravirus salinus infects amoeba, not humans, not fish, not marine mammals. But the particle itself is striking. It's roughly 1 micrometer long, large enough that some bacteria are smaller. Under electron microscopy, it appears as an ovoid shell with an opening at one end. The genome is where the real surprise sits. Pandoravirus salinus carries a double-stranded DNA genome of about 2.47 million base pairs. It encodes around 2,500 predicted genes. The unsettling part is that approximately 93% of those predicted coding sequences had no recognizable match in any existing database when they were first analyzed. Those unmatched sequences are technically called orphans, short for open reading frames without identifiable homologs. The phrase unknown genes does not mean alien DNA. It means that the genetic vocabulary of this virus is largely absent from the records, biology has built so far. The technical process behind the find followed a now familiar route. Researchers collected sediment, placed it with acanthamoeba cultures, and watched for amplification. Once the virus replicated, particles were isolated, purified, and sequenced. What emerged on the inside of those infected amoeba was unusual morphogenesis. Pandoravirus particles assemble through a stepwise process inside the host, rather than through anything resembling bacterial-style division. That detail matters because giant viruses blur the older textbook boundary between viruses and cellular life. They are not cells, but they carry genomic complexity that pushes against the simple definitions. Knowledge of Pandoravirus was lost in a different way than Pithovirus. It was not frozen, it was simply invisible, hidden in marine sediment beyond the reach of pre-genomic methods. The mainstream theory views Pandoraviruses as giant DNA viruses with deep evolutionary histories that diverged early from other viral lineages. A more historical reading treats coastal sediments as preservation archives, layering biological records from changing marine ecosystems over geological time. A fringe view suggests that the oceans may contain entire viral lineages that do not fit any current biological category, lineages we cannot detect because our reference libraries do not yet contain them. The practical reality is that marine viromes, the full viral populations of seawater and sediment, remain poorly mapped compared with land pathogens. Each new sequencing run pulls more oyster fans from the dark. The ocean stores information at genetic scale, even when humans cannot yet read it. From a virus that almost no one needs to fear directly, the story shifts to one whose victims are easy to see and harder to ignore. Number 3, Chelonid Herpesvirus 5. The virus turning sea turtles into walking tumors. Picture a green sea turtle surfacing off the coast of Oahu. Its shell glints in the light. Then it turns and the head comes into view. Tumors crowd the area around its eyes. Smaller growths swell along the mouth and neck. A flipper drags slightly, weighed down by another cluster of tissue. The disease has a clinical name. Fibropapillomatosis, usually shortened to FP. The viral agent associated with it is Chelonid Herpesvirus 5, often written CHHV5 or CHV5. Sea turtles are ancient marine reptiles. Their lineage stretches back over 100 million years. They survived the same mass extinction that ended the non-avian dinosaurs. Today they live in coastal systems shaped by pollution, runoff, boat traffic, and habitat loss. HV5 appears alongside a set of cauliflower-like tumors that grow on skin, eyes, mouth, neck, and flippers. The growths are not subtle, some reach several centimeters across. Eye tumors can effectively blind a turtle, removing its ability to find food and avoid predators. Mouth tumors interfere with feeding and can cause slow starvation. Flipper tumors reduce swimming efficiency, which lowers escape speed and forage range. Internal tumors have also been documented in kidneys, lungs, and the heart, which tells researchers that the disease is not strictly a surface condition. Prevalence is the part that startles new researchers entering the field. In some Hawaiian green turtle populations, FP rates have reached up to 92%. Hotspots include Hawaii, Florida, Puerto Rico, Texas, and other warm coastal regions, particularly those with high nutrient runoff. The technical picture combines a herpes virus infection with a host immune response and environmental cofactors. EOTRC5 DNA is frequently detected in tumor tissues using polymerase chain reaction methods. Yet the virus alone does not appear to be the entire explanation. Many turtles carry CHHV5 without developing FP. The current model suggests that disease expression requires a combination of viral presence, immune stress, and environmental pressure, such as pollution, warmer water, or biotoxins from algae blooms. Knowledge of FP was actually lost in reverse. Tumors were observed in turtles long before viral or environmental causes were understood. Early naturalists documented growths in the 1930s without any way to test for the underlying viral signal. The mainstream theory is that CHHV5 is central to FP, with environmental stress acting as a trigger for disease expression. An archaeological historical reading notes how modern coastal development, agricultural runoff, and habitat compression have changed the disease landscape for turtles whose evolutionary lineage predates almost everything around them. A more speculative view holds that polluted coastal ecosystems may be generating new disease patterns where chronic stress recruits ordinary viruses into devastating outcomes. modern relevance is concrete. Rehabilitation centers across Florida and Hawaii perform tumor removal surgeries, monitor recurrence, and run habitat restoration programs in parallel. The takeaway is that when immunity, pollution, and viruses align, tissue growth itself becomes a trap. And once a virus can derail a turtle that has survived 100 million years of ocean change, the next question is, what happens when a virus targets something even more delicate, the nervous system of a dolphin? Number 4. Cetacean Morbillivirus, the brain disease that strands whales and dolphins. Beaches along the United States Atlantic coast became, in 2013, a long line of evidence. Bottlenose dolphins washed ashore from New York down through Florida. Some were already dead. Others were alive but disoriented, swimming in shallow water, beaching themselves repeatedly, even when refloated by rescue crews. Dolphins and whales rely on extraordinary internal systems. Navigation runs on echolocation. Social structure runs on coordinated calls and memory. Hunting runs on speed, sound, and group strategy. A disease that interferes with any of those systems is not simply uncomfortable, it is catastrophic. The cause of the 2013 event was Cetacean Morbillivirus, or CEMV. The virus belongs to the Morbillivirus group, the same broad family that includes measles in humans and canine distemper in dogs. Its hosts include dolphins, porpoises, and several whale species. The pathology is layered. CEMV targets the respiratory tract, immune tissue, lymphoid organs, and the central nervous system. Pneumonia is one major cause of debilitation. Encephalitis, inflammation of the brain, is the most disturbing effect. When brain tissue swells under viral pressure, orientation, behavior, and social coordination break down. A dolphin with encephalitis can become unable to use echolocation effectively. It can lose the ability to keep up with its pod. It can drift toward shallow water and not understand why. Outbreaks have a documented history. Between 1990 and 1992, a major CEMV event killed thousands of striped dolphins across the Mediterranean. Between 2013 and 2015, the United States Atlantic event recorded more than 1,600 bottlenose dolphin strandings. The National Oceanic and Atmospheric Administration declared the event an unusual mortality event in 2013. Secondary infections played a significant role. Once the immune system is suppressed by morbillivirus damage, bacteria and fungi can move in and finish what the virus started. Environmental factors enter the picture as well. Marine pollutants such as polychlorinated biphenyls, abbreviated PCBs, accumulate in dolphin tissue over time. Researchers studying tissue samples from stranded animals have proposed that these pollutants may suppress immune function and amplify the impact of viral infection. Knowledge of CEMV followed a familiar pattern. Strandings were visible long before virology could explain them. Coastal communities saw dolphins washing up for centuries without a viral framework to interpret what they were seeing. The mainstream theory holds that CEMV directly drives respiratory, immune, and neurological disease in cetaceans. The archaeological historical theory notes that industrial pollutants and coastal change appear to have worsened outbreak vulnerability, especially in long-lived species that accumulate contaminants over decades. The fringe speculative theory suggests that the high social intelligence of dolphins, with its constant close contact and acoustic communication, may make viral spread more efficient through a pod behavior, essentially turning social bonds into transmission routes. A virus does not need teeth to beach a whale. It only has to disturb the nervous system. And while CEMV moves through populations that have lived with it for some time, the next virus in the dossier is something newer and arguably more alarming because it is still learning how to use marine hosts. Number 5H5N1, avian influenza in marine mammals. The bird flu that wiped out 17,000 elephant seals. On the long pebbled beaches of Argentina's Peninsula Valdez, the 2023 breeding season turned into a graveyard. Southern elephant seals haul out there in massive numbers each year to give birth and nurse pups. By the end of the season, the beaches were lined with dead pups. The cause was a strain of highly pathogenic avian influenza A, subtype H5N1. H5N1 began its career as a bird-adapted influenza. For years, its mammalian crossovers were rare, and usually dead-end infections, meaning the virus could infect a mammal but rarely spread to another. That pattern has changed. The current clade of H5N1 has infected an expanding range of mammals, including seals, sea lions, dolphins, otters, and porpoises. The Peninsula Valdez outbreak provided one of the most dramatic numbers in the modern marine mammal disease record. More than 17,000 southern elephant seal pups died in 2023. Estimates of pup mortality reached 95 to 96% in affected colonies. Pup mortality at that scale removes an entire year class from the population. For a species that takes years to reach breeding age, the demographic damage compounds over time. Clinical signs in affected marine mammals include lethargy, neurological symptoms such as tremors and disorientation, respiratory distress, and collapse. H5N1 in mammals can damage lungs, brain, and multiple organ systems. The dense breeding colonies that make elephant seals so visually striking also act as transmission amplifiers. Hundreds or thousands of animals share a small stretch of beach. Respiratory secretions, contact, and shared space all increase opportunities for viral spread. The most pressing scientific concern is mammal-to-mammal transmission. Genetic evidence from infected seals and sea lions has suggested patterns of spread that are not easily explained by repeated independent bird-to-mammal spillover. Some viral genome sequences appear consistent with sustained mammalian transmission chains, although researchers continue to debate the strength of that interpretation. The geographic scope extends well beyond Argentina. South American sea lion die-offs have been documented across Peru, Chile, Argentina, Uruguay, and Brazil. Detection of H5N1 in Antarctic and subantarctic wildlife marks an ecological escalation. Polar regions that once stood apart from major influenza outbreaks are now part of the map. Knowledge of avian influenza was long focused on poultry and wild birds. The shift toward mammalian outbreaks has forced surveillance systems to expand. Carcass testing, environmental sampling, and genome sequencing now apply across species lines. The mainstream theory holds that H5N1 spills into mammals repeatedly and in some cases can transmit among them under the right conditions. The archaeological historical theory traces modern spread to human poultry systems, wild bird migration corridors, and ecological pressure that bring birds and mammals into closer contact. The fringe speculative theory suggests that dense marine mammal colonies may act as evolutionary training grounds, places where the virus encounters new selection pressures and can refine mammal-adapted variants. Modern relevance is direct. Wildlife agencies, public health authorities, and aquaculture operations now run coordinated surveillance with strict carcass handling protocols and warnings against contact with sick or dead wildlife. Evolution rewards every successful transmission chain, regardless of which species it crosses. From influenza, the next virus comes from another familiar group, a Morbillivirus relative that found its own marine niche. Number 6. Phocine Distemper virus, the dog disease that crossed into the sea. In 1988, the North Sea changed character. Harbor seals along the Danish, Dutch, German, and British coasts began dying in numbers that no one had predicted. By the end of the outbreak, more than 23,000 harbor seals were dead. The cause was identified as a previously unknown morbillivirus, soon named Phocine Distemper virus. Abbreviated PDV. PDV is a relative of canine distemper virus and, more distantly, of the measles-like viruses that infect other mammals. It is not simply ordinary pet dog distemper. It is a distinct marine-adapted morbillivirus with its own genetic signature and host range. Harbor seals are especially vulnerable hosts. They haul out in dense groups on sandbars, beaches, and rocky shores. Those gatherings create exactly the conditions that respiratory viruses exploit. PDV causes fever, pneumonia, immune suppression, seizures, disorientation, and death. Many of the visible symptoms overlap with those of canine distemper in unvaccinated dogs. In 1988, the immunological naivete of European harbor seal populations meant that few animals had prior antibodies. Mortality climbed rapidly. The pattern repeated in 2002 when a second major outbreak killed more than 30,000 harbor seals across the North Sea and surrounding waters. The interval between outbreaks suggests that population immunity wanes over time, leaving each new generation vulnerable when the virus returns. Other species play roles in the system. Gray seals appear to suffer less severe disease but may act as carriers or bridge hosts that help maintain the virus in marine ecosystems. Arctic seal species have been studied as potential reservoirs in colder waters. The connection to climate is direct. Research by Tracy Goldstein and colleagues has linked changes in Arctic sea ice to shifts in seal movement and contact between previously separated populations. When sea ice retreats, animal movement corridors change. Populations that rarely met in the past may now overlap. Viruses inherit the new geography. Phocine Distemper virus DNA and antibody evidence have been detected across multiple pinniped species, with patterns consistent with broad circulation across Arctic and subarctic populations. Knowledge of PDV was lost in the same way that many marine pathogens have been hidden. The ocean once concealed outbreak data because carcasses drifted offshore, scavengers removed evidence, and stranded animals were rarely tested. The mainstream theory is that PDV spreads through pinniped contact networks, especially during haul-out events and breeding aggregations. The archaeological historical theory considers how changing human activity, climate pressure, and ecological disruption have altered seal distributions over the past century. The fringe speculative theory suggests that melting ice may unlock future disease exchanges across entire polar basins, exposing previously isolated populations to viruses they have never encountered. Barrier-shaped disease maps. When ice disappears, viruses inherit new roads. From large mammals to something far smaller, the next section moves into the world of aquaculture, where viruses meet industrial-scale farming for the first time. Number 7. Ostreid Herpesvirus. One microvariant, the herpesvirus collapsing global oyster reefs. In 2008, oyster farmers along the French Atlantic coast began watching their crops die. The species at the center of the collapse was the Pacific oyster, Crassostrea gigas, also known by some taxonomists as Megalana gigas. Within days of the first signs, juvenile oysters in affected beds were dying in numbers that no traditional pathogen pattern could explain. The cause was a microvariant of Ostreid Herpesvirus 1, abbreviated OSTV1 microvariant. This is an oyster herpesvirus. It is not a human herpesvirus, and it does not infect humans. It belongs to a group of herpesviruses that infect invertebrates, and its host of greatest concern is the Pacific oyster. The disease it drives is called Pacific Oyster Mortality Syndrome, or POMS. In severe outbreaks, juvenile mortality reaches 80 to 100%. Death can occur within 8 to 10 days of the first symptoms. Larvae, spat, and juveniles are far more vulnerable than adults, which makes the disease especially damaging for hatcheries and grow-out farms that depend on young stock to maintain production. Visible symptoms include gaping of shells, weakness, reduced feeding, and rapid collapse. In larvae, signs include impaired swimming and feeding failure. Transmission moves through water. Infected oysters shed viral particles into surrounding seawater, and OSHV1 DNA can be detected in water samples around dying beds. The microvariant designation refers to genetic differences distinguishing the strain from earlier OSHV1 variants. The microvariant is generally associated with higher virulence and broader geographic spread. Temperature plays a sharp role. Outbreaks tend to intensify when water temperatures climb above roughly 16 to 18 degrees Celsius. That threshold matters because warming coastal waters bring more days each year above the trigger temperature, expanding the seasonal window for outbreaks. Geographic spread has been rapid. After the initial French outbreaks, OSHV1 microvariant has been detected in Australia, New Zealand, the United Kingdom, Ireland, Spain, and other oyster-producing regions. Each new location followed roughly the same pattern: a first warning season, mass juvenile mortality, and an industry forced to adapt. Knowledge of oyster mortality predated the microvariant pattern. Farmers had recorded summer die-offs for decades. What was missing was the specific genetic identification of the variant driving the most severe modern outbreaks. The mainstream theory holds that OSHV1 microvariant drives mortality under favorable environmental conditions, particularly warm water and high stocking density. An archaeological historical reading notes that the global oyster trade has long moved stock between regions, helping spread vulnerable lineages and pathogens together. A fringe speculative view holds that warming seas may select for faster, more aggressive aquaculture viruses, essentially rewarding strains that can complete their life cycles in shorter, hotter windows. Filtration connects oysters to the sea. The same biology that lets an oyster feed and grow also exposes it to viral water. Once an outbreak begins, the medium itself carries the disease. And once a virus shows what it can do in shellfish farms, the logic translates with terrifying clarity to vertebrate aquaculture. Salmon, in particular. Number 8. Infectious salmon anemia virus. The orthomyxovirus that broke Chile's salmon industry. Between 2007 and 2010, the southern coast of Chile became a case study in industrial disease collapse. Chile's salmon industry had grown into one of the largest in the world, with sea cages anchored in cold coastal waters and stocked with farmed Atlantic salmon. The virus that broke that system is infectious salmon anemia virus, abbreviated ISAV. ISAV is an aquatic orthomyxovirus. The orthomyxovirus family also includes the human influenza viruses, but ISAV is not human influenza and does not infect humans. The shared family membership matters because both groups carry segmented negative sense RNA genomes. Segmentation allows reassortment, the swapping of genetic segments between strains, which is one of the key mechanisms behind influenza variation. ISAV infects farmed Atlantic salmon, Salmo salar. Clinical signs include anemia, internal hemorrhage, lethargy, darkened coloration, and high mortality. Internal examination reveals pale gills, an enlarged spleen, abdominal fluid called ascites, and hemorrhage in major organs. Two protein features matter most for virulence. The viral hemagglutinin esterase protein and the fusion protein together determine how efficiently the virus attaches to and enters host cells. A key genetic marker is the highly polymorphic region, or HPR. HPR-deleted strains are generally linked to virulent disease. HPR0 forms, which retain the full region, are less virulent but epidemiologically important because they may serve as ancestors or reservoirs for the more dangerous deleted variants. Chile's major outbreak began in June 2007 on a farm in the Chiloé region. From that initial detection, the virus spread through the closely packed cage networks that define the region's industry. Economic losses reached around $2 billion United States dollars. Approximately 20,000 jobs were affected. Entire fjord systems were temporarily emptied of farmed salmon. The mechanics of spread fit a textbook pattern for dense aquaculture. High stocking density, frequent fish movement between sites, shared equipment, and cage proximity all multiplied transmission opportunities. Knowledge of ISAV existed before the Chilean outbreak. The virus had been documented earlier in Norway and other salmon-producing countries. What was lost in Chile was operational caution. Industry expansion outran the biosecurity and surveillance systems needed to manage a dense farming network. The mainstream theory holds that virulent ISAV spread through dense aquaculture networks once a virulent strain entered the system. The archaeological historical theory notes that industrial scaling created conditions that natural salmon ecosystems would not sustain, with stocking densities far above anything found in wild populations. The fringe speculative theory suggests that aquaculture itself can act like a viral accelerator for marine pathogens, providing both abundant hosts and short transit distances between them. Density lowers the distance a virus must travel. And density, in modern marine farming, is not a marginal feature. It is the entire economic model. The next section follows the same logic into shrimp aquaculture, where the timeline of collapse is even faster. Number 9. White spot syndrome virus. The shrimp killer that can erase a pond in days. A shrimp pond that looks healthy on Monday can be effectively dead by the end of the week. The agent behind that timeline is white spot syndrome virus, abbreviated WSSV. WSSV is a large enveloped double-stranded DNA virus. It is classified in the family Nymiviridae, genus Wispovirus. Its genome is a large circular DNA molecule of roughly 300 kilobases with some variation between isolates. The host range is broad within crustaceans. Pinnate shrimp, prawns, crabs, crayfish, and a wide range of wild crustacean carriers can be infected. The clinical picture is straightforward. Affected shrimp show white cuticular spots embedded in the shell, reddish body coloration, lethargy, and reduced feeding. Shrimp often gather near the edges of ponds in the days before collapse, a behavior familiar to farmers as a warning sign. Mortality reaches 90 to 100% in affected ponds. The speed is what defines the disease. death can sweep through a pond in 3 to 10 days. For a farmer, that timeline leaves almost no opportunity for intervention. WSSV emerged in Asian shrimp farming around 1992 with early reports from Taiwan, China, and Japan. From there, it spread across Asia, into the Americas, and into other shrimp-producing regions. Transmission moves horizontally through water, cannibalism of dead or dying shrimp, contaminated sediment, and infected wild carriers. Hatcheries play a critical role. Movement of infected post-larvae from hatcheries to grow-out ponds can introduce the virus into otherwise clean systems. Wild crustacean reservoirs make the source difficult to see. Crabs and other crustaceans living in or near pond water can carry WSSV without dying themselves, which complicates tracing and control. Knowledge of WSSV evolved through the 1990s as researchers, including those at the National Taiwan University, the Universidad Nacional Autónoma de México, and the Centro de Investigaciones Biológicas del Noroeste in Mexico, worked to characterize the virus, map its hosts, and develop diagnostic tools. The mainstream theory holds that WSSV spreads through contaminated water, animal movement, and farm-to-farm transfer of stock and equipment. The archaeological historical theory situates the outbreak in the rapid global expansion of shrimp farming, which scaled faster than disease management could mature. The fringe speculative theory considers whether shrimp farms may unintentionally synchronize outbreaks across entire coastlines, with shared water sources and shared larval suppliers creating regional vulnerability windows. In water, contact is not only touch. It is the medium itself. Every drop of pond water is a potential pathway. And the same principle that makes oysters vulnerable to OSHV one applies, with different mechanics, to the section's final virus, the one humans actually meet most often at dinner. Number 10. Norovirus in shellfish. The human pandemic that lives inside oysters. A raw oyster platter looks clean. The shells are heavy, the flesh is bright, the presentation is expensive. The platter can also be carrying a viral payload that no inspection can detect by eye. Norovirus is one of the leading causes of acute gastroenteritis worldwide. Most viewers will have either had it or known someone who has. Symptoms include vomiting, diarrhea, abdominal cramps, nausea, and dehydration. The incubation period is short, usually 12 to 48 hours. Duration is typically 1 to 3 days, although shedding of viral particles in stool can continue for longer. The infectious dose is famously low. As few as around 18 to 20 viral particles can establish infection in a susceptible person. That number matters because it explains how norovirus moves so efficiently through cruise ships, hospitals, schools, and restaurants. Transmission is fecal-oral. The route can involve surfaces, food, water, and contaminated hands. The ocean enters the story through sewage. Sewage-contaminated coastal water carries human norovirus particles into bays, estuaries, and shellfish beds. Oysters, clams, and mussels are filter feeders. A single oyster can process tens of liters of seawater per day, drawing in plankton, organic particles, and any viral material suspended in the water. Norovirus particles concentrate in oyster digestive tissues, particularly the midgut and digestive gland. The concentration effect is the key risk factor. Even seawater with relatively low viral contamination can yield shellfish with high viral loads after enough hours of filtration. Contaminated shellfish look, smell, and taste normal. There is no visible warning. Heat destroys norovirus reliably, but raw oysters skip the heat step entirely. That preference for raw consumption is why oysters are repeatedly linked to outbreaks. Depuration, the process of holding shellfish in clean water before sale, is useful for some contaminants. For viruses, depuration is far less reliable. Norovirus binds in shellfish tissue in ways that resist quick clearance, and standard depuration times often do not eliminate viral particles. Sources of contamination include sewage outflows, storm runoff, septic system leaks, and discharge from boats. Heavy rainfall is a known trigger. Storms wash surface waste into coastal waters, raising the viral load in shellfish beds. Knowledge of the route was lost in the same way many foodborne pathways were lost. People blame bad seafood long before viral testing traced the actual chain from sewage to filter feeder to human stomach. The mainstream theory holds that shellfish bioaccumulate human norovirus from contaminated coastal water. The archaeological historical theory traces the increasing pressure of coastal urbanization on shellfish beds, with denser populations producing more waste and stressing aging sewage infrastructure. The fringe speculative theory suggests that climate-driven rainfall extremes may increase future shellfish contamination events as more intense storms overwhelm treatment systems more often. Filtration is powerful. It cannot choose what it collects. 10 viruses in, the patterns begin to repeat. Preservation, concentration, density, movement. The next sections look at the deeper structure that connects all of them. The ocean is not one viral environment, it is many layered archives stacked against each other. Permafrost runs along the northern edge, holding ancient microbes in suspended chemistry. Coastal mud builds up in slow horizontal layers, preserving genetic material across geological time. Reef tissue stores symbiotic and pathogenic viruses inside the cells of corals, fish, and invertebrates. Animal bodies act as living archives, with viral DNA woven into host genomes through ancient infections. Farm ponds and aquaculture cages function as a different kind of archive, recording the viral consequences of human food production. Cold preserves viruses differently than warm seawater. In permafrost, biological information can sit for tens of thousands of years with minimal degradation, as the Pithovirus case demonstrated. In warm coastal water, viruses face faster degradation, but greater opportunities for replication. Sediment serves as a long-term storage medium. Heavy particles settle, accumulate, and bury viral material under successive layers. Pandoravirus came out of exactly that kind of archive. Many marine viruses are difficult to detect until a host or sequencing method reveals them. Amoeba co-culture, the technique used to find Pithovirus and Pandoravirus, works because amoeba are broad hosts that can support a wide range of giant viruses. Polymerase chain reaction methods amplify specific viral DNA sequences from samples, allowing detection of animal viruses such as COBY5 in turtle tumors or SEMV in dolphin tissue. Metagenomic sequencing pulls the entire genetic content of an environmental sample, viral and otherwise, and matches it against reference libraries. The mismatched fraction, sequences with no known relatives, has been informally called viral dark matter. Orphans, open reading frames without identifiable homologs, populate that dark matter and complicate evolutionary interpretation. Researchers cannot easily reconstruct the history of a gene that has no known cousins. Water movement transports viruses across invisible boundaries. Currents carry particles between regions. Runoff delivers viruses from land into estuaries. Permafrost thaw drains ancient microbial material into northern seas. Animal migration moves viruses inside hosts that may travel thousands of kilometers. Dead animals often disappear offshore. Carcasses sink, drift, or are consumed by scavengers, hiding the true scale of outbreaks from beach-based observation. Shellfish convert dispersed contamination into concentrated exposure, turning low background viral loads in seawater into high viral loads in food. The mainstream theory holds that marine viral diversity is massive and severely undersampled. Estimates of total viral particles in the ocean reach numbers that exceed the count of stars in the observable universe by many orders of magnitude. The archaeological historical theory treats old ecosystems as leaving biological traces in frozen and sedimentary records, traces that modern sequencing is only beginning to recover. The fringe speculative theory holds that the ocean may contain viral lineages unlike anything currently cataloged, lineages that cannot be detected because the reference libraries needed to recognize them do not yet exist. Modern science reads nature by following chemistry, sequencing, and host response. Innovation in metagenomics, environmental DNA sampling, and global pathogen surveillance is gradually shrinking the dark matter fraction. The question that follows naturally is what happens to those archives when the conditions that maintain them begin to shift. Warming does not invent viruses, it rearranges their opportunities. The shift is visible across nearly every section of this dossier, even when the connection is indirect. Permafrost thaw provides one of the clearest examples. Frozen ground holds microbial material in chemical stasis. As thaw advances, that material drains into rivers, coastal waters, and seas. Pithovirus is a single, dramatic data point in a much larger ecological process. Sea ice loss reshapes marine mammal movement. Tracy Goldstein and colleagues at the University of California, Davis have published research linking Arctic sea ice reductions to changing seal movement corridors and to the spread of Phocine Distemper virus. Populations that rarely overlapped under historic ice conditions now share waters and contact zones. Warming oyster waters shift outbreak thresholds. The temperature trigger for Ostreid Herpesvirus 1 microvariant outbreaks around 16 to 18 degrees Celsius occurs for more days each year in warming coastal regions. The disease window expands as the climate climbs. Storm runoff increases contamination. More intense rainfall events, consistent with broad climate projections, drive more sewage into shellfish beds, raising norovirus loads after major storms. Host range shifts expose immunologically naive populations. As species follow food, temperature, or breeding sites into new areas, they encounter pathogens their immune systems have never met. Naive populations often suffer the highest mortality rates in early outbreaks. Marine mammals following prey may enter new disease networks they did not historically share. Breeding colonies and haul-outs serve as outbreak amplifiers. Dense aggregations create the close contact conditions that respiratory and contact-transmitted viruses exploit. Elephant seal mortality from H5N1 and harbor seal mortality from PDV both relied on this dynamic. Warming coastal waters stress shellfish immune systems, while changing oxygen levels and salinity in aquaculture systems compound the pressure on farmed species. Disease often emerges from multiple pressures rather than one cause. Fibropapillomatosis in green sea turtles illustrates the principle. Chronic environmental stress combines with ETHHV5 infection and pollutant exposure to produce visible disease. Climate can also synchronize outbreaks across regions. When entire coastlines pass through a temperature threshold at the same time, multiple farms or wild populations may begin failing in parallel. Ice once acted as a biological barrier. Its retreat removes a barrier that biology has used for millions of years. The mainstream theory holds that climate change alters host movement, survival, and pathogen exposure. The archaeological historical theory notes that historic disease maps were partly maps of climate boundaries, with certain pathogens limited to specific temperature, humidity, and ice conditions. The fringe speculative theory holds that Arctic thaw could reveal pathogens absent from modern immune history, creating exposure events without recent precedent. Forecasting, sentinel monitoring, and climate-linked disease models are now active research areas in institutions ranging from the World Organization for Animal Health to academic ocean labs. Disease ecology follows thermodynamics, movement, density, and time. From climate, the next section focuses on a more concentrated factor: industrial-scale aquaculture, where human design choices act as direct viral amplifiers. The contrast between wild ocean spread and farmed ocean concentration is the central feature of modern marine disease. Wild populations are dispersed. Farms are not. Aquaculture packs hosts into predictable, repeated, high-density systems. The Pacific oyster industry, as discussed under OSHV-1 microvariant, depends on hatcheries that produce billions of spat each year and grow-out beds that hold them in closely spaced trays. Salmon farming, as illustrated by ISAV, uses sea cages that can each contain hundreds of thousands of fish. Shrimp farming, as illustrated by WSSV, depends on ponds that hold dense post-larvae populations through a short, intensive grow-out cycle. Juveniles are often more vulnerable than adults across these systems. Oyster spat, salmon smolts, and shrimp post-larvae all show higher susceptibility than older animals in many disease scenarios. That vulnerability concentrates at exactly the life stage where farm density is highest. Hatcheries are critical control points. Infection introduced at a hatchery can spread through stock movement to dozens or hundreds of grow-out sites. The pathways out of a hatchery are also pathways for a virus. Fallowing is one disease control strategy. By leaving a site empty for a defined period, farmers break host availability and allow viral particles in water and sediment to degrade or disperse. Zoning is another approach. Regulatory zones limit the movement of stock and equipment between sites, reducing the chance of farm-to-farm transmission. Surveillance testing before moving animals is now standard in many regions, particularly for ISAV in salmon and OSHV-1 variants in oysters. Selective breeding for disease resistance has produced new shrimp and oyster lines with better outcomes against specific viruses. The Australian and French oyster industries have invested significantly in breeding programs aimed at OSV1 tolerance. Antiviral treatment is rarely practical in marine farming. The volumes of water and the cost of pharmaceutical-grade antivirals make medication an unrealistic strategy at industrial scale. Water exchange is both necessary and dangerous. Farms need water flow for oxygen, waste removal, and temperature control, but water flow also delivers viral particles between sites. Cages and ponds do not fully isolate animals from surrounding ecosystems. Net cages exchange water with the open sea. Pond walls leak. Wild carriers move in and out. The financial scale of losses provides a useful frame. The ESAV outbreak in Chile produced losses of approximately $2 billion United States dollars. The cumulative history of WSSV losses across the global shrimp industry is estimated in the tens of billions of dollars. The mainstream theory holds that density and movement drive aquaculture viral outbreaks more than any other factor. The archaeological historical theory traces the rapid global expansion of seafood demand and notes that industry growth outran disease management systems. The fringe speculative theory holds that industrial coasts may function as artificial evolution chambers, where high host densities and frequent disease pressure select for faster and more aggressive marine viruses. Biosecurity, genetic resistance, vaccination where feasible, and lower density production are the central tools for reducing risk. Farming the sea means obeying the same biological limits that govern wild ecosystems. From aquaculture, the next section asks the question viewers care about most. Which of these viruses can actually reach people? Not every virus in this dossier represents a direct human health threat. The clearest direct risk is Norovirus. Raw shellfish can deliver human viruses concentrated from sewage-contaminated water. Outbreaks have been documented across dozens of countries, often linked to specific harvest areas after heavy rainfall or sewage overflow events. H5N1 in marine mammals is a surveillance concern rather than a current pandemic threat. The virus does not yet spread efficiently from seal to human. The reason it matters for human health is that each new mammalian outbreak provides opportunities for genetic changes that could, in some future strain, support more efficient human transmission. Mammal adaptation matters for influenza risk in a way that is well understood from past influenza pandemics. Pithovirus and Pandoravirus infect amoeba. They do not infect humans. Their danger is scientific uncertainty about what else may be preserved in similar archives, not proven human disease. Wildlife workers, rehabilitators, fishers, and aquaculture farm workers form higher exposure groups. Anyone who handles sick or dead marine wildlife may encounter viruses such as H5N1, CEMV, or PDV more often than the general public. Carcass handling is a documented risk in H5N1 response. Protective equipment, including masks and gloves, is now standard guidance during marine mammal stranding events involving suspected influenza. Marine mammal strandings more generally require protective protocols regardless of which virus is suspected, because secondary bacterial and fungal infections can also pose risks. Seafood safety follows a few clear principles. Cooking shellfish to appropriate internal temperatures reliably destroys norovirus and most other foodborne viruses. Appearance does not reveal contamination, which is why regulators rely on harvest area monitoring rather than visual inspection. Wastewater treatment is a public health barrier. Modern sewage treatment systems remove a substantial fraction of viral particles, but no system removes 100%, and aging infrastructure or overflow events can release untreated waste directly into coastal waters. Environmental monitoring can detect viral contamination before outbreaks. Wastewater surveillance, now widely used for SARS-CoV-2, has expanded to include norovirus and other targets, allowing early warning of community-level circulation. Foodborne illness, wildlife conservation, and pandemic surveillance form three distinct categories of risk that often involve overlapping pathogens but different policy responses. The mainstream theory holds that human risk varies sharply by virus and by exposure route, and that specific behaviors such as raw shellfish consumption or wildlife contact carry disproportionate weight in total risk. The archaeological historical theory notes that human settlement near coasts has always linked waste, food, and waterborne disease, with the same pathways visible in archaeological records of medieval port cities and modern urban estuaries. The fringe speculative theory considers whether expanding coastal pressure may create new crossover opportunities as more people, more livestock, and more wildlife share shrinking coastal zones. One Health surveillance, the coordinated monitoring of human, animal, and environmental health, is the current framework for integrating these risks. Safety comes from understanding pathways, not from fearing the ocean as a whole. With that framework in mind, the last section steps back to consider what all 14 previous sections add up to. Return to the first image, a virus preserved for 30,000 years, beginning to replicate inside an amoeba in a French laboratory. That image connects, in some structural way, to every section that followed without repeating their details. Oceans do not merely contain viruses, they organize viral opportunity. Preservation runs through cold permafrost, deep sediment, and host tissue. Pithovirus survived in frozen ground. Pandoravirus survived in sediment. Endogenous viral elements survive inside animal genomes themselves, encoded into the DNA of hosts that absorbed ancient infections over evolutionary time. Amplification runs through dense colonies, farms, reefs, and shellfish beds. H5N1 exploited dense elephant seal colonies. PDV exploited harbor seal haulouts. OSHV, one microvariant, ISAV, and WSSV exploited industrial farms. Norovirus exploited filter-feeding shellfish to concentrate viral particles from dilute coastal water. Movement runs through currents, migration, trade, storms, and melting ice. Viruses ride hosts, water, equipment, and weather systems. The same currents that distribute nutrients distribute pathogens. The same trade routes that move oysters and shrimp around the globe move their viruses with them. Vulnerability runs through stress, pollution, warming, and immune naivety. Stressed turtles develop fibropapillomatosis. Polluted dolphins suffer worse outcomes from CEMV. Immunologically naive seal populations die in larger numbers during PDV outbreaks. Each pressure compounds the others. Viruses succeed by exploiting existing physical and biological pathways. They do not invent new ones. They follow what is already there. The ancient scale of Pithovirus, measured in tens of thousands of years, sits in the same dossier as the industrial speed of ISAV and WSSV, which can collapse a farm in days. The invisible genetic mystery of Pandoravirus sits alongside the visible tumors of green sea turtles. The neurological disease of dolphins sits alongside the foodborne illness of human shellfish consumers. The deadliest viruses, in the broader sense used here, are not always those with the highest human fatality rate. Deadliest can mean ecological loss, economic collapse, animal suffering, or crossover potential. By that definition, an oyster virus that erases reefs may be as significant as an influenza variant that threatens future pandemics. The mainstream theory holds that ocean virology is entering a genomic surveillance era, in which metagenomic sequencing, environmental monitoring, and One Health frameworks reshape what humans know about marine pathogens. The archaeological historical theory holds that past ecosystems stored viral information that modern science is only beginning to recover, and that the record of life includes more chapters than current biology has read. The fringe speculative theory holds that unknown marine viruses may outnumber known viral threats by orders of magnitude, and that humans encounter only a thin surface layer of the actual marine virome. Modern relevance now spans sequencing, wildlife monitoring, aquaculture reform, climate modeling, and wastewater control. Each tool addresses a different part of the same system. Fear is a poor instrument for understanding any of this. Sharper observation is a better one. Innovation, in the practical sense, begins when humans stop treating nature as empty background and start treating it as a structured environment that obeys consistent rules. Viruses obey those rules with no exceptions: contact, replication, preservation, and chance. The ocean gives them all four.